Hi everyone. Today we will be discussing about secondary structure of protein. Uh, so, if we assume like how protein folded in a particular pattern to do some function. So first there is some amino acid sequence. This amino acid sequence are aligned to get one polypeptide stretch, and this polypeptide is folded in a particular secondary structure, and from different kind of combination of secondary structure we get one tertiary structure, and if it's required, various tertiary structure interact to form one quaternary complex, right? Now we can compare it with one basic example, that's the word sentence or linguistic model. So there is word, from word we can get sentence, from sentence we can get paragraph, from paragraph there should be some uh, chapter, and then finally one book, right? Now we consider a word as amino acid, then the sentence as the polypeptide chain or 1 degree structure, the paragraph that's the secondary structure, the chapter that's tertiary, and book is the final quaternary structure, right? Now uh, in case of we have to focus on secondary structure, and there are three different kind of secondary structure. One is alpha helix, another is beta sheet, and the third one is the turn. Okay, in maximum time it's beta turn. So the first concept of protein secondary structure comes in 1951. Okay, and the first paper was published in PNS by Corey. Okay, Pauli and Corey, and that's why they were awarded Nobel Prize. So that at that time there is no such kind of development of X-ray crystallography and cryo-EM, right? So it's quite a fantastic idea at that moment that they got. Now, if we see the how two amino acids are connected to form one peptide bond, so there suppose alanine is there, glycine is there. Now we know there is some kind of covalent bond formation that is called peptide bond, right? And how the peptide bond is formed? So suppose there is CH3NH, then CH here, then there is CH3, then CO, then NH, and here it should be CH and H as its glycine, and then it's CO. That so this is N terminal, this is C terminal, right? And there are two R group. Here this is a methyl, and here is only hydrogen bond. Now, if we zoom it, uh, we get multiple dihedral angle. So that here there are three different kind of dihedral angle. One is phi, another is psi, and another is omega. That's generally used. So the dihedral angle between C and N and C alpha is known as phi. So that angle we can write here phi, and the angle between C alpha and C is known as psi, and the angle between that CO or NH we can write it as omega. So here's a similar thing, we can write here as psi, we can write here as phi. So now I would like you to focus here. This is phi, this is psi. So phi is the angle between N and C alpha. Psi is the angle between C alpha and C, and omega is the angle between uh, CO or NH and CO. Basically the C and N. Okay, now there should be some different combination of phi, psi, and omega. So omega has two things. One is 180 and another is 0. So maximum time it's 0, that indicate that it's in Cs, and 180 means it is in trans conformation. So what can be the possible value of phi and psi? Do you think that it can be anything, right? Minus 8, 180 to plus 180? But it's not possible, because there are huge number of steric hindrance, right? Different kind of R groups are there. That's why the phi and psi is restricted. And based on the restriction, there are different phi and psi value that's already determined by different conformational technology like X-ray crystallography or something. And Professor G. N. Ramachandran first observed that there are only particular region is allowed between of the area of phi and psi. So here the allowed area is indicated with some color, so blue color, deep blue. Okay, and the white region is not allowed at all, and that light blue area indicating that, yeah, that allowed, this is allowed, but some modification should be done to incorporate this particular stretch. Okay, so that's the Ramachandran plot. Here the phi and psi value are plotted. And why this value are plotted? Because the steric hindrance will not allow the amino acid to take any kind of phi and psi value. So there is some limited phi and psi value. That's why uh, we should plot the phi and psi value in that particular plot. Now we'll focus on alpha helix. Why alpha helix form more radially than the other possible conformation? It should come in your mind that there is some kind of helical structure, and to uh, ensure that the space is quite limited to design, you should design some kind of conformation. You can design alpha helix, beta plated seed to arrange the particular stretch of amino acid. But why alpha helix is quite more common? Because it from intra-strand hydrogen bond, intra-strand hydrogen bond, and the intra-strand hydrogen bond is formed between I and I plus 4 residue. So the I here, that's the CO carbonyl group, and the NH of I plus 4, that from the hydrogen bond here. And here one imaginary axis is assumed, and then the polypeptide length is just wrapping around the axis, right? So that's called one pitch. Here you can see that's called one pitch, and one pitch consists of 3.6 amino acid. And in case of alpha helix, the distance between two alpha carbon or two amino acid is 1.5 angstrom. So there are 3.6 amino acid residue on average, and 1.5 angstrom is the distance between two amino acid. They will get 1.5, 1.6, sorry, 1.5 into 3.6, that is 5.4 angstrom. It's here, and that's the top view of alpha helix. So all the R group is basically outside from the helix region. You can see here that's out. Okay, everything. And in case of top view also, that's outside. Now this is a similar thing here, and you can decide which one is the left-handed and which one is the right-handed by some thumb alignment rule. Okay, now not every amino acid has the same propensity to form alpha helix. So some amino acid has more propensity to form alpha helical structure, and if we see that alanine has the highest propensity to form alpha helical structure, and some amino acid like asparagine, cysteine, and proline, like they're quite rare in alpha helical structure. So, and also this charged residue, like if we take some polyglutamate or polyhistidine, this poly stretch of some charged residue, due to repulsion they can't form the alpha helix. And some the presence of proline and glycine is also not favorable, because proline contain the immuno group, so it can destabilize the alpha helix, and glycine is quite flexible. So if we take 20 or 10 stretch of glycine, it will form some random coil instead of making some uh, alpha helix, right? So you should remember which amino acid should be selected. So it basically depends on the bulky hydrophobic group or the exact hydrophobic group that will help to stack the amino acid and to form a particular to maintain a particular shape of alpha helix, right? Now the another thing is beta strand, and here the two side chains are just outside of just outside of this particular plane. And uh, if we see two different possible possible here, two different combination. So this is NC and this is CN, right? And the distance between two amino acid is 7 angstrom here, and the hydrogen bond that is formed here, it's inline hydrogen bond, so that's quite preferable. And in case of parallel, the hydrogen bond is not inline, and the orientation is here from N to C and N to C. Okay, the same orientation, and the distance is 6.5 angstrom. It's quite shorter than the antiparallel. If you see the stability, the antiparallel seat is quite more stable than the parallel seat, because of the inline hydrogen bond formation. Okay, and to connect the things, we need some kind of turn, and here three different turn is possible. One is alpha turn, one is beta turn, one is gamma turn. In case of alpha turn, it generally consists of 4 residue, 5 to 4. So this is I and I plus 4. The hydrogen bond is formed between this residue, and that's formed between I and I plus 3, 4 residue. That's between I and I plus 2, 3 residue, almost 3 residue. This is 4 residue, and this is 5 residue. That's all about today, and generally the beta turn is more preferable. The proline and glycine is used to present at I plus 1 and I plus 2 location, and this beta turn generally present in some anti-parallel strand to make it more favorable. Okay, so that's all for today, and yeah, we'll continue.